Hello students, a warm welcome to all of you in our 7 days online certification course on topic NGS. NGS is a next generation sequencing. This is the first session of this training program. In today's session, we discuss about the overall idea of NGS. We discuss about what is sequencing, what is the types of sequencing, what are the generation of sequencing, means basic idea about the sequencing. Okay. Now we start today's session and first we understand what is sequencing. What is the sequencing? Sequencing is the process by which we easily determine the exact order of building block. We easily determine the exact order of building block. Building block of biological molecule. These biological molecules may be your DNA, maybe your RNA, maybe your protein. Okay. The building block of protein is amino acid. Whenever we find out the exact order of amino acid, then this sequencing technique is known as a protein sequencing. Okay. Whenever we find out the exact order of, okay, and the building block of DNA and RNA is nucleotides. Okay. Block of nucleic acid is your nucleotides. Whenever we find out the exact order of nucleotide, then this is known as a nucleic acid sequencing. Okay. Then we divide this nucleic acid sequencing into two parts. First is your RNA sequencing and second is your DNA sequencing. DNA sequencing means whenever we find out the exact order of adenine, thymine, guanine and cytosine in your gene in any genome, that is known, then this technique is known as a DNA sequencing. Okay, the DNA sequence is the laboratory technique that used to determine the exact order of nucleotide, and this nucleotide may be adenine, may be thymine, cytosine and guanine in your DNA molecules. Okay, this sequence encodes the genetic information that directs develop or functioning of all the living organisms. Okay. Now, what is the application of this DNA sequencing? This DNA sequencing plays a most important role in field of biology as well as in field of medicines. Okay. By the help of this sequencing technique, scientists can easily determine the gene. They easily study about the genetic variations, explore how the gene is expressed and regulate. And by the help of this sequencing technique, we easily and also diagnose the genetic disorder, also develop the target therapies, and also plays a most important role in personalized medicines, in drug designing or discovery, and drug discovery, and also plays a most important role in vaccine design. Okay. Now we understand the history behind this sequencing. Okay. First, alanine tRNA is sequenced. Okay. First, this alanine tRNA is First, this alanine tRNA is sequenced via Robert in 1965, and the sequence of alanine alanine tRNA is 77 nucleotides. This there is 77 nucleotides. Okay. After this 1967, after this 1965, in 1970s, a new sequencing technique is also developed, that is the maximum Gilbert sequencing. By the help of this sequencing, we easily find out the exact order of DNA. In this technology, we use some hazardous compounds. Okay. This technology is also known as a chemical degradation sequencing method, because here we use some chemical compounds to degrade or to find out the exact order of DNA. Okay, but in this technology, in this sequencing method, we use the hazardous compound. That's why this sequencing is not so much successful. Okay, or not widely used. But in after the 70s, in 77, a new sequencing method is developed that is known as a Sanger sequencing, and this sequencing method is developed by the Frederick Sanger, and they also find the Nobel Prize for this technology, and this is a gold standard for the 30 years. Presently, we also use the Sanger sequencing method, okay, for sequence the short nucleotide sequence. This is the limitation of the sequencing method. Okay, short sequence, short DNA sequence. Okay, this is the drawback of the sequencing technology. In this technology, we design only we sequence only short sequences or short DNA. Okay, in this Sanger sequencing, we also known as this sequencing method is also known as a chain termination method. Okay, here we use dinucleotide triphosphate to terminate the chain. That's why this sequencing method is also known as a chain termination method. Okay. After the 1977, in 2005, a new era is started, that is known as a NGS, that is the next generation sequencing. Okay. First sequencing is your pyro sequencing or 454 sequencing. Okay. Then in 2006, Illumina sequencing is developed. Then the solid sequencing is developed, and iron tolerant sequencing is developed. Iron tolerant sequencing in 2010, solid sequencing in 2007, and Illumina sequencing is 2006. Okay. Then after the 2010, the new era is generated, that is known as the third generation of sequencing. Here we include the sequencing method like SMART or PacBio. This is developed in after the 2010 and Oxford nanopore sequencing. Okay, presently, most of the time we use next generation sequencing and Illumina sequencing. Okay, and this is a little bit history about the sequencing. Okay, now first we discuss the first generation of sequencing. In this first generation of sequencing, we included the two types of sequencing method. First is your maximum Gilbert sequencing, that is also known as a chain termination sequencing. Next is your Sanger sequencing, that is also known as a chain termination sequencing. Okay, in this sequencing, first we prepare the library. then sequencing, then gene electrophoresis, find out the exact order of sequence, order of your molecule. Okay, which amplify the template DNA and gel electrophoresis emerged to approximately same time, but completion time is different. Okay, this technology is have lots of advantages as well as some disadvantages. Like in maximum Gilbert sequencing, we use some hazardous compounds, and this is very harmful for humans as well as environment. Okay, but in chain, the Sanger sequencing is safe, but it is a very time consuming and expensive sequencing technology, and this is efficient for and this is not appropriate for the large genome. This is appropriate for the small genome, small size of genome. Okay, in human genome project, whenever we find in human genome project, we sequence the genome of the organisms by the help of this Sanger sequencing. Okay, now now we discuss some little bit idea about the Sanger sequencing. Okay, now we discuss about the second generation. Now we discuss about the second generation of sequencing, that is known as the NGS, and this is the main topic of our internship program. Okay, this next generation sequencing evolutionizing the DNA sequencing by massively parallelizing the process, allow the millions of DNA fragments to be sequenced simultaneously. Okay, in the sequencing technology, we use Illumina sequencing, 454 pyro sequencing, and intolerant sequencing. What is the Illumina sequencing? We discussed in detail. Okay. Now we discuss 454 sequencing, that is also known as a pyro sequencing. Okay. What is the principle behind this pyro sequencing? Whenever the nucleotide sequences is added into the growing template synthesis process, during this process, two phosphate molecules are released out, and this two phosphate molecule is known as a pyro sequence or pyrophosphate. And here we focus this pyrophosphate. That's why this sequencing method is known as a pyrosequencing. Okay, whenever this pyrosequence is released out, then it will emit some light, and this emitted light is detected by the detector. Okay, let's suppose this is a growing chain. Now a new nucleotide is bind. During this, whenever this new nucleotide is bind, then this pyrophosphate is released out. Whenever this pyrophosphate is released out, then this will also emit some light, and this light is detected by the detector, and by detecting this light, we easily determine which types of amino acid is added. Okay, this is the basic principle behind this pyrosequencing. Next is your intolerant sequencing. In the sequencing, we focus the pH change. Okay. Whenever a new nucleotide is added into the growing chain, the pH of the media will change. Okay, and we target this changing media by the detector. Okay, this pyrosequencing higher read length than the Illumina sequencing become a, and the cost of this pyrosequencing is also very high. Intolerant sequencing is a faster and cheaper but has a high error rate in homopolymer sequencing. Okay. Now we discuss the advantages of NGS. Advantages at NGS, it is a high throughput technology. It is a cost effective for the whole genome sequencing and high accuracy. But disadvantage is it is a short read length, approximately 100 to 300 base pair. Okay. But For interpreting the data of NGS, you require a complex bioinformatics tools. Like in normal window, we can't analyze this data. To analyze this data, we require a Linux environment. That's why if you have Linux in your PC, if you are Linux, then it's okay. If you have Windows, then you first create a subsystem of Linux, then you interpret this data. Okay, how this process will perform, we will discuss in our incoming session. Okay, this is the drawback of NGS. Now we discuss the third generation of sequencing. Third generation of sequencing this sequencing technology also allow single sequence of DNA in a real time without need any amplification. Okay, because this is also an advanced technology. This is also high throughput technology. Okay, first is your PacBio sequencing. It observes the real time polymerase incorporation. Okay, this technology and this sequencing method is very appropriate long reads, and this is very useful for the repeated reason. Okay, next is your Oxford nanopore sequencing. In Oxford nanopore sequencing, we target the electric current change. Whenever the nucleotide sequence is bind, then this current will change, and we detect this current changes in current by the help of detector. In this technology, we pass the DNA molecule through a nanopore. Okay, whenever this nanopore is passed to this nanopore and the new nucleotide will add, then there are some variation in current, and this variation of current is detected by the detector. Okay, this is a portable, fast, but error rate is very high. Okay, what is the advantages? Advantages is a long read length, no amplification required, and this is a faster sequencing technology. Okay, disadvantages, it is higher and this is very expensive. Okay, this is the third generation of sequencing. In first generation of sequencing, we include the maximum Gilbert sequencing as well as Sanger sequencing. In second generation of sequencing, we include Illumina sequencing, 454 sequencing, and Eintolden sequencing. In third generation, we include PacBio, Smart sequencing, Oxford sequencing, etc. Okay, these are the three generations of sequencing. Now we discuss what is NGS, what is the types of NGS, what is the workflow of NGS, etc. Okay, what is this NGS? NGS is a high throughput technique, and this is a modern sequencing method that allow the rapid sequencing of entire genome, transcriptomics, and target region of DNA as well as RNA. Okay. Unlike the Sanger sequencing, which sequence the DNA fragment at a time, NGS can process millions of fragments simultaneously. This NGS is very fast, cheap, and more comprehensive as compared to Sanger sequencing. Okay. This is very fast, cheap, and comprehensive. That's why it is known as a high throughput sequencing, because at a time it will process and it will sequence millions of fragments. Okay. Now these are the different types of sequencing. Like first is your Illumina sequencing, Eintolden sequencing, 544 pyro sequencing, PacBio Smart sequencing, and Oxford sequencing. Illumina sequencing sequences while synthesized using the reverse terminators. We discuss this in detail. Okay, but read length is 75 to 300 base pair. Disadvantage, it is very high accuracy and high throughput. Eintolden detect the hydrogen ion released. Basically pH change, we already discussed. Read length 100 to 400 base pair. Very fast and cost effective. Pyro sequencing, Pyrosequencing means here we focus the pyrosequencing means light emission whenever the pyruvate are released out, then light is emitted and this light is detected by the detector. Read length is 400 to 700 base pair, longer read. PacBioSmart, single molecule real-time sequencing, and read length is very large, like 10 to 15 kilobase pairs. Long reads and detect modification. Oxford nanopore, we measure the electrical current changes, and length up to megabase pair, kilobase pair or megabase pairs. Okay, this is portable and real-time sequencing. These are the different types of sequencing, their principle and their advantages and read length. Okay. Now we discuss the advantages of NGS. Advantages of NGS: it is very high throughput sequencing, means per run we sequence millions of base pair. This is very high sensitive and accuracy, cost effective, can detect low frequency variant and suitable for the diverse sample like DNA, RNA, FFPA and cfDNA. Okay. Okay. FFPA means formalin fixed paraffin embedded DNA. Okay. Next is cfDNA means circulating cell-free DNA. Okay, means by the help of this sequencing, we easily sequence the paraffin embedded DNA as well as circulating cell-free DNA. Okay, but there are some limitations of this NGS. Means it requires the advanced bioinformatics tools, short read length as compared short read length, high initially cost, and potential wise in amplification and library preparation. Okay, these are the limitations. Now we understand the workflow. First step is your isolation of DNA and RNA. Isolation of DNA or RNA, which is your target molecule. Then fragmentation means we fragment out this DNA into the small fragment. Then add some adapter at both 3' side and 5' side. Then PCR. Okay. Then our library is prepared. Then we check the quality of our library by the help of TAP station, quiescent and bioanalyzer. TAP station is the automatic electrophoresis technique. Okay. Then clonal amplification means each fragment is amplified via the bridge amplification on a cell tube. This is a type of cell tube, and in this cell tube a bridge amplification is processed. What is the bridge amplification? We discussed in detail. Okay. After the bridge amplification, we apply the sequencing method. And the sequencing method may be Illumina, maybe ion tolerant, maybe PacBio, maybe nanopore. Most popular is your Illumina sequencing. Now, after the Illumina sequencing, we find out the data. Okay. Then we read or Then we find out the data in FASTQ file. Okay. Then we check the quality of this data by the help of a software that is known as a FastQC. Okay. Quality check by the help of FastQC. Then we alignment. For alignment, we use a software like HISCAT2, VWB and VT2. Then we feature count. Then we count the feature. Then we count the feature of our DNA. For this, we use a software that is a feature count. How we perform blast, how we perform the alignment, how we perform the feature count, we will discuss in our incoming session. Then for the visualization, we use IGB or UCSC genome browser. UCSC genome browser is a type of database. We also use RStudio. And to interpret this data, RStudio also plays a most important role. Okay, this is the overall workflow of NGS. Okay, this is the overall workflow of NGS. In next session, we discuss this workflow in detail. Okay, now we discuss the application of NGS. Application of NGS. First and wider application is whole genome sequencing. Okay. Okay. Second application in whole exome sequencing. Third is target gene panel sequencing. Okay. Next in transcriptome sequencing or RNA sequencing. Next in metagenomics and epigenomics or small RNA sequencing. Okay, single cell sequencing, mitochondrial genome sequencing to find out the Y sequence of Y chromosome, animals and plants genome sequencing to find out the ancestry relationship in microbiology to find out the genome of microbial strain, monozygotic twins, microRNA, and to interpret the data like gene interpretation, STR interpretation, mixed strain interpretation, and multiple STR. Okay, this is the application of NGS. Okay, now we discuss how we retrieve the NGS data from NCBI SRA. Okay, so we understood this. Open the Google. Then in Google we simply search NCBI SRA. Okay, then open this home page of NCBI SRA. This is the home page of NCBI SRA. Okay, here this is the all databases. These all are the databases we present in NCBI portal. Okay, this is the documentation, how to submit, how to search downloads, how to use SRA in cloud, submit the SRA, number of softwares, on this page we perform number of tasks. Okay, now we search our target molecule. Like my target molecule is E. coli. Now I simply search. This is my target molecule. Now this is the, now this is the result. Means in NCBI 4838424 and E. coli related data is present. Okay. Means in NCBI 26055 data is present related to the E. coli. Here the access is public. These are the different options. We also apply this filter according to our requirement. Like total access is public access is the 26051. Sources is your DNA and RNA. DNA is 24843, RNA is 877. Genome type is genome. Library layout is paired and single, and these are the platform. Means by the help of this sequence is generated like capillary, helicose, Illumina, iron tolerant, LS54, Oxford, PacBio, etc. But our target is Illumina. Okay, here the strategies you want: epigenomics, exons, genomics, and other. These are the clouds. Then these are the file you want: PEM file, FASTA-QC file, which you want. And here you also apply the organism, apply the filter for the organisms, and this is the data like biosample, bioproject, data GPA, and GEO databases. Okay. And this is your search detail. Now I apply the E. coli in all fields. Now let's suppose I want to apply the filter. Let's suppose I want to search the data related to the Illumina. Now I simply click on this Illumina. Now this is the data related to the Illumina. These are the data from Illumina sequencing. Okay, this is the complete data. Okay, now I want to remove out this filter. To removing this simple filter, simply click on clear all. Whenever you click on this clear all, then all this filter are delete out. Okay, now I select the molecule. Now I select this one. Okay, now open this one. This is the SRA ID, this is the whole description, this is the external ID, this is the submission detail, this is the study, this is the sample, and this is the library, and this is the running spot, and your size is 22.2 MB. Okay, this is your ID, spots, basis, size, and published. Now I open this one. Whenever you open this page, then this page will open. Here you find the number of information, like for this is the meta information, run, spot, base pair, size, GC content, public, and their experimental information, biosample information, and their bioproject information. Then in next, this is the information about the analysis, about how many identified, unidentified, DNA, RNA, all the information. Then this is the information about the reads. Then this is the information about the data access, SRA normalized, light, version, create, location, name, etc. Then here the option to download the file. This is the accession number. This is the number of base pair total. And here you download this file into FASTA format as well as FASTA-Q format. I want to download this result in FASTA-Q format. That's why I click here. Okay. Now the starting is start. Now the sequence is downloaded in my PC and it will take time. Okay, and this reads is loading. Okay, wait a second. Now this data is downloaded in my PC. This is the data. Okay, and this data is downloaded in zip file. Okay, how to unzip this file we discussed in next session. Okay, in this way we in this way we easily retrieve the NGS data from NCBI SRA. Okay, here is a limitation. If your data is less than 2 GB, then you easily download this. If your data is more than 2 GB, then you require SRA toolkit. Okay, this is the limitation. Okay, this is the way, this is the overall idea, and this is all about the today's session. In today's session, we discuss the overall idea of sequencing, and we also discuss how we predict the data from NCBI SRA. This NCBI SRA is a database of, it is a database of NGS, that is the next generation sequencing. Okay, and this is all about today's session. Thank you so much for watching my video. Thank you.